Yes,
So
we're here.
thank you, James. And I also like to thank Jen and the ALS ONE committee for inviting us to share our latest findings. And it is my great honor and privilege, as you said, to co-present here today alongside Professor Cudkowicz and share key elements from Paradigm, our phase 2b study. So let's start. But first, forward-looking statement, as we are a public company, but that won't stop us. Okay, so the pathophysiology of ALS, as we all know, is very complex, and it's underlying multiple pathways and mechanisms that are involved. And this is why we believe that we need to shift the paradigm and not to think about the solution as a silver bullet rather than a combination therapy. So Prime-C, our lead candidate for ALS, is a tablet assembled of 2 FDA-approved drugs, celecoxib, which works on the neuroinflammatory process as a COX-2 inhibitor, and ciprofloxacin, who is a fluoroquinolone affecting regulation of microRNA, and also iron metabolism, as it's also an iron chelator, in which each one of the components targets, as you can see, several of these key pathologies. But importantly, the unique extended release formulation of Prime-C really allows us to maximize the synergies between these two components. And in demonstrate the effect and the synergy between these two components, we conducted a preclinical studies utilizing zebrafish. We had 2 key models for that in collaboration with 2 laboratories using TDP and SOD1 mutant. And as you can see in front of you, when performance of these animals, we clearly see a beneficial effect for each one of the components and a clear synergistic and significant effect for the combination. Here we can see that relative to riluzole, which was the standard of care. And also we see the same effect when we are looking on TDP-43, the other model. And not only that we saw this significant effect in the motor performance, but we also looked on the effect on the synapses. And here, when looking on the connectivity, as you can see on the right panel, we see the same pattern. So we see a significant effect for the combination. And this effect was also demonstrated not only in the neurons, but also in the supporting cells, such as the microglia. And this study was published in 2020. And we moved forward from that and reinforced these results using the same methodology. Again, each one of the components and then the combination. Here we utilize iPSC's model in a great collaboration with Justin Ichida, who presented here beautifully yesterday. So here again, we looked on the survival of the neurons using blood from ALS patients. Again, we see a clear effect of each one of the components and a significant beneficial effect for the combination, as you can see in the light blue. And by focusing just on the 12 months for our convenient, as you can see here on the right panel. So again, we see beneficial effect for each one of the components and a significant effect for the combination. Actually, here it was similar to the healthy control. Yeah. Next, we also wanted not only to show the efficacy, but also to try to demonstrate the target engagement of the drug. And for that, we utilized our phase 2A study. It was an open label study with 12 patients over 12 months of dosing. This work was done in collaboration with Mass General using the NILS biodepository samples. Together, it was a 3-stage biomarker study. So at first, we characterized utilizing neuronal-derived exosomes at a set of around 12 markers that relates to our target engagement, but also relates to the disease, such as TDP-43. So we had a cross-sectional study just to demonstrate the baseline effect of each one of these markers. Here we have 2 representative, TDP-43 and prostaglandin 2, which is the downstream marker for the neuroinflammatory process. So we had a cross-sectional effect, and then we chose samples from the biodepository, which are characterized according to a long 12 months effect. And lastly, we measured the effect on our 12 months treated samples. And as you can see really nicely, following 12 months of dosing with Prime-C, we saw a significant reduction in TDP-43 and in accordance to that, we also saw a significant effect on prostaglandin 2. And these results, together with the supporting findings that we obtained from the preclinical studies, advance us to our phase 2b study paradigm, which we'll be focused on today. And Merit will tell you all about that.
Thank you, Sharon, and welcome everybody to this great ALS ONE symposium. So the paradigm study was a phase 2b study. It enrolled participants with ALS who were relatively early in their illness. 73 people were screened, and it was a 2-to-1 randomization to active drug versus placebo for 180 days. And then the option for participants to continue an open label extension. And we're going to show results of both the randomized control period as well as the open label extension up to 12 months. Main outcome measure is safety and tolerability in and ALS-related biomarkers. Secondary measures included looking at ALS functional range scale, breathing, a quality of life questionnaire, and complication-free survival. One important thing I want to note is that at the baseline, the groups were well balanced. Here we just show the ALS-FRS at baseline, which was the same in both groups, but this is true for the other key baseline characteristics as well. Next slide, please. So the majority of patients did go in the open label extension. So first I want to just show safety and tolerability. Big picture, the drug was very safe and tolerable. The treatment emergent AEs were similar in number between the active and the placebo and the double blind. And there were slightly more studied drug-related TEEs on active drug. These were typically mild and GI-related and resolved. And they were also anticipated, known from the drug. So overall, good safety, both in the double-blind period and the extended open label extension. Next slide, please. So I'm going to show you some of the secondary outcome measures, and here starting with ALS functional range scale. So blue is prime C, gray is placebo, showing the rate of change of ALS-FRS score. And you see already at 4 months, a statistically significant difference between the groups favoring prime C. And then at the 6 months, it's 29.2% difference. So slowing of the progression after 6 months. And the p-value at 6 months is 0.12, again, driven by the sample size of the study. And you can see if you look at change from baseline, that it's a 2.23 difference between active and treatment. And that's, an important function preservation for people living with ALS. Here we see the 12-month data. So you see as people go into open label extension, you can go to the next slide. That difference remains with a 36.6% difference, a slowing of the illness in people who started prime C early compared to those who started at 6 months later, again suggesting a disease modifying effect, and the point difference is 6.56. Next slide. So we saw effects on all parts of the ALS-FRS. Yeah. I don't see you going to the next slide, but yeah, there we go. So we saw it on all the domains, but it was most pronounced on speaking and swallowing or bulbar function. But we saw that people on drug compared to placebo in all 4 of the domains had better outcomes on ALS functional range scale. Next slide, please. So we looked at other outcome measures. We saw the same positive trends across all the secondary exploratory endpoints, including survival. Here again, we see the double blind and the open label extension data, and we see that same benefit of prime C on slow vital capacity, the different staging approaches, the Kings and the Mitos. There fortunately were not many mortality events, so we don't have enough events to weigh in on that, and also on quality of life. Next slide. So we've also looked at some of the biomarkers, where this is still a work in progress. But I wanted to show you a few that we have. As you heard from Sharon, prime C works on iron-related dysregulation. So we did see here that Prime C restores transferrin and ferritin. And so we know that in people with ALS, elevated ferritin and lower transferrin are correlated with a more rapid decline in ALS-FRS score and a faster disease progression. And we do see that again in the placebo participants, but we see that Prime C induced a normalization of ferritin and transferrin, and that was again related and associated to attenuation of disease progression. So really an important biomarker effect of Prime C, and really demonstrates the target engagement that was expected. Next slide. These are also early data and more to come here. But Prime C also had a significant impact on key ALS microRNAs. Again, this was expected based on the mechanism of the drug and the preclinical work. But there were some key ones that that had a significant impact on. So to date, it shows targeted modulation of over 24 microRNAs, and we didn't see that in the placebo group. It demonstrated more than 700 significantly differentially expressed microRNAs, where in the placebo arm there were 80. Some key ones, it looks like it engages miR-199 A-5P, which is important for regulating neuroinflammation and neuronal survival, and also miR-151A-5P, which is associated with oxidative toxicity, and the last one, miR-126-5P, that's related to motor neuron degeneration. Again, there is more work to be done on these microRNAs that will be presented at other conferences, including the International MND meeting in Montreal. So this is a collaboration with Professor Noam Shamran. So I think that's my last slide. Oh no, just some key summary things. Very favorable safety and tolerability profile, achieving the primary endpoint, which was safety. We saw with Prime C improved disease progression as measured by ALS Functional Range Scale by 29.2% after 6 months, and also had reached statistical significance at 4 months. We also saw people who started early versus people who started later of the drug, a 36.6 improvement in disease progression. We saw this effect on all ALS-FRS domains. We saw potentially better survival at 12 months. Again, the numbers are small. We saw consistent positive trends on all the clinical outcomes, and we saw target engagement in both in markers of iron dysregulation, including transferrin and ferritin, along with a significant impact on regulation of key microRNAs that are associated with ALS progression. So it suggests a disease-modifying effect, good safety, and really a good data to move forward to the planned phase 3 trial that NeuroSense is working on. And just to give you like a big picture of what that design is going to look like, it's going to be a international trial, double-blind, placebo-controlled trial with an open-label extension. It's going to look at safety and efficacy. The primary endpoint will be a joint assessment of function and survival at 12 months. Obviously, we'll look at different safety measures. We're going to take people with ALS in the trial, about 300 people, and it'll be it's planned to be in US, EU, Canada, and Israel. We're planning a 2-to-1 randomization, and again, we're hoping that the first patient will be enrolled in the first half of 2025. Next slide. So just a huge thank you for the people that were part of this study and the entire team at NeuroSense. We wouldn't have this phase 2 data to really guide the phase 3 without all their participants. Thank you.
Wonderful. Thank you so much. Well, really terrific to hear about the program from start to finish. We do have some questions that have come in. Let me just start sort of at the beginning. What was the choice? I think this is, Sharon, maybe one for you. What drove the choice to study zebrafish, and have you looked at mice? And if you did look at both zebrafish and or mice, did you see the same readout in ferritin or iron, or is that even something you can look at in zebrafish?
Yeah, so it's a great question. Being in the field of preclinical studies, I think that we all know that all models are wrong, but some are useful. So I would say that back then, we knew that there was a huge study with celecoxib using the SOD1 mice, which actually didn't show that it was predictive in terms of going from preclinical studies into clinical studies. And back then, the zebrafish really rise as a promising novel model for drug screening. So we know that there is a huge 97% of homology, and it's really show really nicely the effect on the brain and also on the motor performance. So this is exactly why we chose the zebrafish. There is a lot of advantages into that. Basically, they are transparent, so you can clearly see the morphology. You can use large number of samples. So this is kind of what guide us, and also considering the fact that we are using 2 FDA known drugs, it really allows us to move forward directly from zebrafish into clinical studies. So we don't have time to waste in ALS.
Yeah, makes sense. There's a question, perhaps predictably, about whether you've looked at neurofilament light and whether what you saw there.
Sure, I'll answer that first. Yeah.
Okay. Yeah, we have looked at the neurofilaments at 6 months. So after the double blind, we saw a nice trend over there, and we do plan to test neurofilaments at the 18 months period, which is just ended this week. So the plan is to test all time points at the end of the study and to really see the effect.
Okay, great. So more data coming. That's terrific. Great question too. There is a question about whether Prime C may be available as an EAP option.
You know, I hope, you know, I think the most important thing is to get the phase 3 study off the ground and started. And as many know here, there's this terrific federally funded EAP program. And that's something that could be considered to apply for. But it's important to have that concurrent with an ongoing phase 3.
And I think we'll finish with this one, although there are more. Thank you for the very nice talk. Did you see target engagement for celecoxib PGE2 level reduction? And if so, how do you reconcile that with the previous single dose celecoxib study where no target engagement was seen? Are the effects here driven by ciprofloxacin, do you think?
Oh, wow, that's a terrific question that I have so many slides that I want to show you now. But basically, in short, I will just say that in our phase 2A study, we indeed show target engagement by measuring prostaglandin 2 in neuronal-derived exosomes. So basically, we could measure the levels in the CNS in a non-invasive way in the blood. So we did see changes following the treatment. Obviously, this is also something that we plan to do now in our phase 2B study. And to answer the question about why didn't we saw that in the celecoxib alone, this is why exactly we are combining the two together. So we also showed in rodent study that the administration of the two components together actually gain more effect. So we see more penetration to the brain through the MDR1 pumps, and we also see more of the drug. So we really worked hard in showing the effect and the impact of these two components together.
Wonderful. So there are other comments in the chat. I'm not going to be able to get to all of them, but I think it was, just testament to an engaging presentation. So thank you very much.